Good afternoon, everybody, and welcome to our monthly Senior Fellow Society webinar. I'm delighted to introduce and to welcome our guest this afternoon, Dr. John Clark. John is a consultant medical microbiologist at Epsom and St Helier University Hospitals NHS Trust. You will not be surprised to hear that he has spent 2020 and 2021 working in the Trust COVID response team. John has held many important roles in his trust. He has been the lead infection control doctor, he's been director of medical education, and currently is divisional medical director for clinical services. In addition, he has recently been appointed as senior responsible officer for diagnostic in South West London. John has a particular interest in the history of vaccination, and as such, I can think of no one better suited to discuss vaccination from smallpox to COVID than John Clark. Once again, a reminder to post any questions you may have on the chat line. The talk will be recorded and so can be viewed later if required. So many thanks for taking the time to address us, John, and it's over to you. Thank you very much.
Thank you very much. Then hopefully the presentations appeared on the screen.
It has actually, very clear.
Excellent. So thank you very much for asking me to talk to you this evening about vaccines and the rise of kind of the vaccine movement. And I wanted to just make clear that I can't talk about vaccines without talking about the rise of the anti-vaccination movement as well. But what I want to do is really break down the vaccine talk into just a brief overview very quickly about what vaccines are, the types of vaccines, just so we're kind of understanding where we're talking about. And then I really want to spend about half of this talk talking about the vaccine rise and some of the key vaccinologists in history and people that I kind of have a dear attachment to, and then spend the last few minutes briefly talking about anti-vaccination. So, myself here. So if we talk about vaccine, at its crudest, a vaccine is just a biological preparation that you give to someone, and it provides acquired immunity to a specific infectious disease. It's really kind of targeting the adaptive immune system, allowing antibodies to be produced. And the easiest way to classify vaccines now is to kind of have the classical and modern type. The classical, really the ones that we all have kind of grown up with and used to be inactivated, live attenuated toxoid conjugates. And you can see that there's different ways of manufacturing these vaccines, and the kind of key thing for most of these is it takes time. So to create a live attenuated vaccine, you have to do serial passage through something such as chick embryo to develop an attenuated strain of a virus such as measles, and that can take years to develop, and then years to test its safety profile, and then years to roll out. Similarly, inactivated vaccines take time. So these vaccines are the ones that we will be talking about in predominant, but actually when we come to things like the COVID vaccine, these are what we call the kind of the new era, the modern era of vaccines, which are really kind of two groups at the moment, viral vector and the RNA. And the key differences between these two and the previous group is that what we're doing here is we're using our understanding of genetics of RNA and DNA sequences to effectively create the sequences that we will be implanting into cells to effectively allow those cells to manufacture the antigens. So rather than subunit vaccines for Hep B, for example, we actually tell the cells to produce those subunits themselves to create the antigen, which then go to antigen-presenting cells to create the immune response. So it's actually a very neat way of doing things, and the RNA vaccines that we're all now so aware of in terms of Moderna and Pfizer really were developed predominantly to try to act as a vehicle for gene editing, so things like cystic fibrosis, and they have had variable success in that field, but actually it was kind of a leap to say, well, actually we can use this mRNA package to actually just insert mRNA from viral proteins. And so the advantage of these types now, this new type, is that it allows you much more rapidity with developing a vaccine to then roll out into the testing phase. But obviously the thing that's happened with COVID is that we've had kind of a worldwide alignment of testing, and I'll go on to COVID briefly later on. I think it's very important to say that vaccines aim to give protection, but nothing is 100% effective. You can have people that, for whatever reason, just don't produce an immune response or have an inadequate response. And that can be they didn't have enough time before they were exposed to the actual virus from the vaccine or the toxin, or they were infected with a different strain that isn't covered. There are lots of reasons for it, and it's important because, especially when we talk about the rise of the anti-vaccine movement, we do need to understand that vaccines are not this kind of 100% effective medication. And it's also important to realize that all vaccines carry side effects. Most of these are relatively mild, temporary, temporally and located around the injection sites most of the time. But you do have people that have allergic to some constituents within vaccines, and you also have some very rare side effects that can occur with certain vaccines. And again, we know about these, and by and large, these are felt to be clinically justifiable given the huge benefit that the vaccine actually gives. The one advantage of COVID, I will say, is that when it gets to this stage in the talk, I don't need to explain what R number is. It's the reproduction rates for infections, and actually when you look at viruses, I think the key thing to point out is the kind of SARS-CoV-2 is about 2.5 to 3 in terms of its R number, we think, based on the information we have to hand. The kind of flus that we see, the seasonal flus, are normally just over 1. But actually, when you look at things like measles and polio, you start getting into these enormous R numbers. And the reason that's important is the higher the R number, the higher the vaccine coverage needs to be, or the higher the immunity needs to be for herd immunity kicks in. And that will become very important when we get to the anti-vaccination part of this talk. So, vaccines, let's talk about vaccines. I'll start by doing a little bit of interaction. I mean, obviously, this is just a bit of harmless fun. You can think about this yourself wherever you are. Who was responsible for the earliest account of smallpox inoculation? So if you said Jenna, you don't score any CPD points. If you said 16th century Chinese physicians, then by all means give yourself a CPD point. And there are lots of written accounts of smallpox inoculation in China predominantly, and also northern India, where they took smallpox scabs, ground them up, and then either inhaled them or you scratched them into the skin. And so this was kind of the variolation where you took the actual smallpox exudate and gave that high risk associated with lots of problems, risks of infection. So anyway, it's a bit mean. So who was the first to describe smallpox inoculations in the UK? So a chance to redeem and get your CPD point back. So if you said Jenna, I'm afraid you're out of luck. If, however, you said Lady Montague, then give yourself a bonus CPD point. Lady Montague was the wife of the ambassador in Istanbul at the turn of the 18th century.
Sorry,
and at that
John,
time,
John, I'm sorry to interrupt you. Yeah, your sound is cutting out a little bit. I think your microphone might need plugging in properly.
okay,
I'm sorry.
that's all right. Can you hear me now?
Yes, thank you. Sorry about that.
Okay, no, that's fine. I'll try and keep an eye on it. Yeah, so Lady Montague was the wife of the British ambassador in Istanbul around the 18th century, the turn of the beginning of the 18th century. And at that time, there was a large smallpox outbreak. And she had read accounts of variolation occurring in the Far East, and people had brought it along the Silk Road, this practice. And so she had her son inoculated in 1718 in the embassy in Turkey, and was so impressed with the results that she brought that practice, variolation, smallpox inoculation, back with her to England and had her daughter inoculated in 1721. And she actually persuaded the Princess of Wales at the time to have it. And this was then followed up by a study where we had volunteers from prisoners. And whilst it showed that there was protective benefits, there was this risk of infection and death, and actually a number of the prisoners died, although the rest of them were then subsequently immune to smallpox. So again, I've been very sneaky and misled you. So who was the first person in England to describe a case of vaccination using cowpox to prevent smallpox? And so here we've got Jenner. If you said Jenner, then I'm afraid you're wrong. If you said Benjamin Jesty, then give yourself a bonus CPD point. Benjamin Jesty was a farmer in Dorset, and at that time it was well known amongst dairy circles that if you had small cowpox, you did not then go on to catch smallpox. And 1774 arose and smallpox hit his village in Yetminster in Dorset. He actually inoculated his wife and 2 sons with cowpox at great consternation in the village, and his neighbours were not happy with him. But actually none of his family then subsequently contracted smallpox. And it was almost 3 decades later that he actually received retrospective recognition for that. He wasn't a self-publicist, he hadn't really done very much, but he'd written to a medic about his work, and this was finally recognised by a pair of gold lancets that came out of the POC institution. And if you're ever in the area of Yetminster, you can go to the farm and actually see the blue pack. So let's talk about Jenner, because we've been kind of skirting around him, and he's thought of as the kind of first true vaccinologist. He was a George's trainee under John Hunter. He was a fellow of the Royal Society, actually for his work on the cuckoo, where he demonstrated that the chick was the one who chucked the other eggs out, or the other chicks out of the nest. It wasn't the adults, because at the time they thought that the adult cuckoos would come in, lay the egg and kick everything else out. He went on, got his MD at St Andrews, where he became an expert in atrial fibrillation. And it was while he was working that he started to realise and hear these stories about cowpox and its protective effects against smallpox. And as you're probably aware, the famous experiment on the 14th of May, where he inoculated the son of his gardener with pus from the blisters of a milkmaid, Sarah Nelms, which came from the cow Blossom, and he demonstrated that James Phipps was immune to a subsequent exposure to what would have been a lethal dose of smallpox. I'm not sure he would have got away with the ethics of that nowadays, but certainly at the time, he published a paper submitted to the Royal Society, who threw it out as garbage and said, nope, this is not good enough. He went through multiple revisions, which to anyone who's had any kind of academic work done, I'm sure you can share the pain. He did more work, he did extra cases, and then finally submitted it and got it accepted. And that was his original paper in 1798 for the vaccination using cowpox. And as any good academic, why stop with one paper when you can try and get 2 out of it? And so he got one the subsequent year in 1799, essentially the same thing. It is safe to say that these 2 papers, when they came out, set the medical world afire. I cannot emphasize enough how big this was, such that rapidly this information spread across Europe. At the time, Napoleon was currently engaged in a war with England, but he went and had his troops vaccinated, and he awarded Jenner a medal, and basically wrote to Jenner and said, ask and you shall receive. And so Jenner said, can we have our prisoners back, please? And Napoleon did. As I said there, he could not refuse anything to one of the greatest benefactors of mankind. So it was clear that people saw the value in this. And Jenner actually then subsequently went on to receive 2 payments from the Royal Society, one of 10,000 grant and one of 20,000, which was a princely sum back at 1802 and 1807. So around the time of the beginning of the 19th century, we had the first meeting of the Royal Jennerian Society, which Jenner had created with fellow medics. And actually, this is where Richard Dunning, who was a Plymouth surgeon, coined the term vaccination based on the cow. And Jenner, to his credit, said, that's not my word, this is Richard Dunning's, he should be credited for it. And the Royal Jennerian Society grew over the next few decades as a kind of source of learning and sharing of information. And within 2 decades of the kind of rollout of vaccination using cowpox, London saw a reduction in smallpox deaths by over half from what it had been, such that 20 years after that, in 1814, we had the first Vaccination Act, which basically allowed free vaccination for infants, bearing in mind that at the time there was no NHS, you had to pay to get this medical input. So all infants were vaccinated, and it actually went the step of banning variolation because of the risk of spreading smallpox. About 13 years after that, the UK Vaccination Act, so the second National Vaccine Act, came into force, which was much more stringent, mandated smallpox vaccination in infants under the age of 3 months, with heavy fines and potential imprisonment for people who did not have their children vaccinated. And over the next kind of 50 years, the smallpox vaccination just continued to grow around the world. It went to America, it went through into kind of, Eastern Europe and into kind of the Middle East. If you look now to our next big figure in the vaccine history, which is Pasteur. And Pasteur obviously is about 50 years after the Vaccine Act. It was about 100 years after Jenner. And he was predominantly working on animal vaccines. He was identifying and isolating these pathogens. And he was credited with the first laboratory vaccine for chicken cholera, as it's known, Pasteurella morbicida, which was a huge thing for poultry farmers. It meant that half their chicken flock didn't die. A few years later, he managed to successfully attenuate the rabies virus to allow rabies vaccination in dogs. And obviously there's this now famous story of Joseph Meissner, the young child there, who's his picture a year later, who's bitten by a dog, rabid, and he basically offered him by his mother the doses of attenuated rabies vaccine, which was for dogs, don't forget. And actually Meissner survived and was so indebted to Pasteur that he worked as his kind of laboratory odd job man, his caretaker, and even after Pasteur died, he continued to look after his tomb. Such was his gratefulness for basically being cured or protected from rabies at that time, which would have been a death sentence. If you look over the next few years, Pasteur brings out attenuated cholera vaccine and the inactivated anthrax vaccine, again predominantly for livestock, but he was demonstrating repeatedly that you could, if you isolated the organism in laboratory conditions, you could start to produce vaccines against them through these various mechanisms. Then we get to Glennie, a British immunologist, who showed that formaldehyde inactivated tetanus toxin, and he then used that as a toxoid vaccine. And he also is credited with isolating the diphtheria toxoid, the toxin from diphtheria, which then allowed Gaston Ramon, who's a French veterinarian, again veterinarian, to go on to develop an inactivated diphtheria toxin, which obviously went on to save countless lives. And Ramon has one of the distinctions of being one of the few people who has got 155 Nobel Prize nominations, but never actually won. Then we get into the 30s, and this is where the vaccine development and the kind of era of vaccine really goes up a notch, and we really start to see kind of real kind of significant advances. And the first person we talk about is the South African virologist Max Thieler, who ended up working in America, and he develops the yellow fever vaccine. And actually it's a 17D, which is named after the 17th run of culture attempts, and it was the D egg in the kind of chicken embryo techniques, which actually produced a attenuated strain from the wild type, which allowed him then to go on to produce this relatively safe yellow fever vaccine, which effectively offers probably lifelong protection. We initially thought it was 10 years, but it's much longer. And he subsequently goes on to get the Nobel Prize for that a couple of decades later. But he's pivotal because he lays the foundation and raises the profile of vaccine development in America in particular. The World Health Organization really was kind of pivotal for what happens over the next 50 years. But one of the first things they did after the Second World War was they praised this technique that had come out of the Pasteur Institute of freeze drying and vacuum packing, which would allow vaccines to be stored and distributed worldwide with no loss of potency. And that would be really important for their subsequent smallpox eradication drive. You get to Jonas Salk, American virologist, lives in New York, and he had been working on polio for some time and had identified a inactivated, a killed vaccine based on the polio type 1 virus. And he starts a vaccine trial in the US with over 1 million children. Results are published a year later showing it to be 80 to 90% effective against paralytic polio. And it's immediately taken up and rolled out. Polio was an enormous issue, killing thousands of children every year in America, crippling tens of thousands. Interestingly, Salk chose not to patent his vaccine, although he was slightly shunned from medical circles because he didn't actually give due credit to his co-workers. But his vaccine was rolled out, very, very effective. However, just a year after his vaccine had been started to be mass produced and rolled out, there started becoming case reports of children who had been given the vaccine and developing paralysis in the arms that they were vaccinated in, and then going on to potentially cause paralytic polio. And those reports were raised over the next few weeks. And in total, there were 40,000 cases of polio related to the issuing of Salk vaccine, leading to 56 cases of paralytic polio, and 5 of those died. And this was traced to one of the 5 laboratories that was responsible for developing the vaccine, the Qatar laboratory, and it was found that their inactivation steps were not sufficiently robust. What it did do was it caused the polio vaccine program to be delayed. It was postponed from 1955 to 1963, and it's estimated that tens of thousands of children were undue or unnecessarily developed paralytic polio because of this delay. So again, we start to see some of these incidents playing in, which get into the press of adverse reactions related not necessarily to the vaccine itself, but how it was produced. Around the time that the Qatar incident occurs, Hilary Kapowski, a Polish virologist who works in the US, had begun working with attenuated oral poliovirus, live attenuated virus. And he was doing a vaccine trial in the Congo. It was funded by the US government, but actually the US government didn't take this up, nor did they take up the subsequent oral live attenuated poliovirus from Sabin, which I'll come on to in the next slide. Around this time, the World Health Organization pushes for smallpox eradication based on the smallpox vaccine and the ability to store and transfer it. So in 1959, we have Albert Sabin, who used his oral polio vaccine to 10 million Soviet children as the trial. He actually develops a trivalent oral polio vaccine for the 3 strains of polio. And it's wildly effective. he refused to patent his vaccine, and he could have actually patented his vaccine, and actually also refused any financial benefit from the vaccine at all. A true kind of inspiration where, you know, he lived for his job and he just wanted to make sure that this vaccine was as widely available as possible. And it was stunningly successful. Yes, there were occasional reports of reversion to wild type in the bowels of some of the babies, but on the whole, the side effect profile was really quite low. And the difference between this live attenuated oral polio vaccine and the Salk inactivated one was that this created good immunity at the guts, which was the site of polio adhesion and integration into the body, and so actually prevented polio from causing a viremia. And so whilst the Salk protected you from paralytic polio, this actually stopped you from getting polio completely. A few years later, we have John Ender, who again is considered one of the kind of the father figures. He actually did two things. So he got a Nobel Prize for being able to isolate polio virus outside of nervous tissue, and started the whole development of various tissue cultures to allow us to grow viruses in various other tissues other than the ones that they infect. But what we remember him most for, other than that, is developing this Edmundston B strain of measles virus, which is still used to this day as a live attenuated measles vaccine. Again, wildly successful and wildly efficacious. The mid-60s also saw WHO really intensifying the smallpox eradication program, and so they were buying up smallpox vaccine, distributing it to the NGOs and health agencies around the world, and actually paying for people to go out to do the vaccination program. You know, it cost millions, but their goal was to eradicate the smallpox. Only has a human host, it has no natural reservoir, and so it's a prime target. Likewise, polio only has a human host, no natural reservoir, so it's also a prime target for eradication. 70s, early beginning 70s saw the trivalent MMR measles rubella licensed. By the mid-70s, we saw the last case of variola major, the more severe kind of hemorrhagic type illness caused by smallpox, which was in Bangladesh. And then a couple of years later, we had the last naturally, I say naturally occurring case of variola minor, which was thought to be the kind of more milder cutaneous but could still kill. This was Ali Malim from Somalia, who became a bit of a celebrity as the last kind of naturally occurring case. But he wasn't the last case of smallpox. Unfortunately, a year later, we had a photographer at the University of Birmingham by the name of Janet Parker. And she worked in the University of Birmingham Medical School, and she contracted smallpox and died. And everyone was kind of like flabbergasted, you know, where had this come from? But attention quickly turned to the laboratory below her photography studio, where work was being done on smallpox virus. The outbreak ended up with 300 contacts quarantined. Her mother actually contracted smallpox but survived, although was quite scarred. And actually, we could spend a whole talk about this, but the press hounded Henry Bedson, who was the head of the microbiology department, and unfortunately, he was picked as the scapegoat and took his own life because of the stress. And actually, when you look at it, you know, he was made aware by World Health Organization about biological safety changes, and he hadn't fully implemented them. But actually, what he was doing wasn't necessarily wrong. And so he, you know, history is kind of looking back and is slightly more kinder on him than at the time. But it caused a huge ruckus and actually led to a whole raft of health and safety legislation being brought out. Biocontainment was pushed into laboratories and became much more rigorous, and also led the World Health Organization to become much more stringent about who had licenses to continue to work on smallpox. And the work was primarily being done in case of the arrival of a mutant escape strain. Fast forward a couple of years to 1980, and we get the declaration of smallpox eradication. It had been several years since the last naturally occurring case. It's actually a triumph of vaccination that we have completely eradicated a viral illness that devastated and killed millions over the years. And so, it was a real cause for celebration. If you look through the kind of 80s and 90s, World Health Organization really started to work on polio. It was pushing to get polio eradicated. And actually within the mid-90s, we had the last case of polio in the Americas, which was in Peru. And the Americas were declared clear of polio in the mid-90s. And if you look at this picture here, you can see kind of in the 80s, you can see all the red countries that had ongoing endemic polio, and by the mid-teens of the 2014, you can see that really polio has been eliminated from the globe, apart from a couple of countries, one around kind of Sierra Leone, the other one kind of Pakistan, Afghanistan border, where there is kind of still ongoing skepticism and suspicion of NGOs going out with vaccines. But we're very, very close. If we can just get into those areas and again get this vaccine drive going, then there's no reason why polio can also not be eliminated. And again, another massive successful story. And this graph just goes to show you the kind of reported cases annually has dropped down to almost nothing. When you come to the noughties, the kind of big things now is more about kind of labs working together to kind of create stuff. And we had the human papillomas virus quadrivalent vaccine, which again initially came out for teenage girls as a way of preventing cervical cancer, and again has dropped cervical cancer rates quite significantly. Hasn't gone completely, but it has dropped it significantly. 2014 saw the arrival of the Group B meningococcal vaccine, which fills in the gap from the existing meningococcal vaccines to basically now leave us with a complete protection against most of the meningococcal vaccines that cause invasive infections, especially kind of meningitis. And interestingly, the Group B vaccine, the way it works, where it's looking at one of the surface proteins, actually confers protection against most of the other meningococcal types, although it's only licensed for Group B, so it's a slightly silly situation. But we are now at this stage now where we have broad meningococcal cover. And then we get to the COVID vaccine. And the thing to say about COVID was it was built on a lot of the groundwork that had been done for MERS. If you cast your mind back, this was the, don't go to Egypt and kiss a camel, you might catch MERS. It was the DNA-based work, RNA-based work, and viral vex vaccines were already in development for the MERS virus to create a MERS vaccine. We had rapid sequencing of the RNA genome for the SARS-CoV-2, which came out on the 11th of January 2020. That allowed us to utilize these kind of new types of vaccine, where as soon as you know the genetic sequence, you can put that in to actually start creating the string of protein, the mRNA, to then integrate into your vaccine. And currently, as it stands, there's 22 vaccines authorized. 6 of them have got WHO approval based on more stringent standards. And there's a mixture. So we have RNA vaccines, as we all know, the Pfizer one, Moderna. We have the adenovirus vector vaccine. So we have the Oxford AstraZeneca, that's the Oxford group led by Dame Sarah Gilbert. The Sputnik 5, which is the Russian one, which again uses adenovirus, 2 types. The Janssen, which is a single dose, and the Convitia, which is a single dose. We have some of the more traditional inactivated vaccines which were rushed through development. The Coronavac, the Sinopharm Chinese one, the Covaxin, the Indian one. The jury's still out on their actual efficacy. Some of the ones for the Sinovac show that it's 51% effective, which would put it in the realms of no better than saline. Subunit vaccines are still pending authorization, and this was the Novavax trial. So the real key here was that we utilised the new technology to get vaccines out very quickly and did things in parallel. So previously, where you would have a vaccine, you do the lab phase 1, phase 2, and then phase 3. Here they were putting some of the phase 2 and phase 3 in parallel to speed things through. And on the whole, it's been a relatively great success, especially in terms of hospitalisation and death of the at-risk groups. So, a romp through vaccine history. It is very easy to think of anti-vaccinations as being a truly modern American phenomenon of slightly deranged, crazed people who have lots and lots of agendas but want to stop kind of medical fascism. But it's not true. As long as you have had vaccines, you've had anti-vaccinations. It's a classic cartoon from around 1802, based on the smallpox vaccination program, where people are turning into cows. You had the vaccination monster, which was in the papers around the 1800s, where the vaccine cow is being fed poor innocents by these horned doctors who are then kind of sort of spewing out loads of dead babies. And you have the righteous people with their swords of truth, which is the anti-vaccine lobby, coming to do battle with these evil medical practitioners. So for as long as we've had that, you can actually trace the anti-vaccination origin prior to Jenner's work with the cowpox vaccines. This goes back and dates back to kind of Montague and the variolation. The earliest I could find was in the 1772, Reverend Massey, who was a prominent kind of cleric who was doing tours of the UK, delivering sermons on the sinful practice of inoculation. And there was a similar reverend in America. And their kind of crux of the matter was that these pestilences were punishments from God, and therefore man creating a way to circumvent these illnesses were working against God's work and were therefore in league with the devil. And so it was very much a religious, moral stance about doing God's will. And so that was the predominant prevailing mood for kind of the late 1700s into the early 1800s. When you get into the early 1800s, you see this move from this kind of religious opposition into kind of more of a political and legal and kind of liberty stance. And it all kind of really crystallizes around those two laws I mentioned earlier, the British Vaccine Laws, Vaccine Act, and then the Second Vaccine Act in 67. And it was all around the kind of mandatory, this is now a mandated requirement with punishments. And it birthed the Anti-Vaccine League and the Anti-Compulsory Vaccination League. And these were kind of political movements trying to use legal measures to kind of step away from this requirement, this mandatory obligation to do it. And it led to riots and marches, 1,800,000 anti-vaccinations in Leicester. And subsequently, we actually had the Vaccination Act in 1898, which brought into law for the first time this concept of conscientious objector, which would then percolate down through legal circles for years after. We had smallpox outbreaks going on in America at the end of the 19th century. So around the time that we had all the rise of the movements, the anti-vaccine lobbies in the UK, because of these smallpox outbreaks, what would normally happen is you have public health officials would come in and mandate vaccines and quarantine, etc. And so you saw the development of the Anti-Vaccination Society, especially following a visit by this prominent anti-vaccinist, William Tebb, who you'd think, oh my God, anti-vaccinist, he must be a madman. But actually, he co-founded the Royal College, which then became the Royal National Society for the Blind. He was vice president of the National Canine Defence League and very active in the Royal Society for Protection and Cruelty to Animals. But he had very, very strong views on liberty, and actually was fined 13 times to have his third daughter vaccinated. And so we saw the growth of the anti-vaccine lobby and the anti-compulsory vaccine lobby in America. And obviously that then swept across the whole of the Americas. Fast forward 50, 60 odd years. So the anti-vaccine lobbies were still there, but obviously with smallpox becoming less and less prevalent, and the rise of all of these other vaccines that were being developed, what we started to see was reports of other things. And this paper in '74, which was related to Pertussis Inoculation, basically said that they suffered neurological complications and got fits, and they basically were advocating that you shouldn't give it to anyone who had any kind of family history of fits, or anyone with a kind of intercurrent infection or related to kind of neurological problems. However, what happened was it went into the media, and you had the patient advocate groups. So there was an association of parents of vaccine damaged children who were angling to try to get financial compensation for presumed idiopathic illnesses related to vaccines. And what it basically led was this kind of hysteria where for the next 2 decades we saw a slump in pertussis uptake. And as you would expect, the moment you stopped your pertussis vaccine uptake down to 1 in 3, we had major outbreaks of pertussis causing serious illness and deaths in children. And it only really got better when the Joint Committee Vaccine Institute demonstrated after sort of 7 years that there was no correlation with fits, and paid GPs to vaccinate children. But that instilled in a lot of GPs this kind of confusion. What's going to mixed messages? The press are saying this, the JCVI are saying this. What should we do? At the same time, you also had prominent physicians. So Gordon Stuart was a Scottish public health doctor who published reports again about neurological illnesses, and it continued to muddy the water. We had a medical establishment which was kind of almost split, saying, well, actually these things cause harm, and actually no, these things don't cause harm, and the benefits outweigh the risks. And the JCVI actually then ended up doing a multi-decade encephalopathy study, which showed in the 80s that there was no real risk of neurological deficit from diphtheria. But it took years to undo this. Briefly talk about thimersals. This was a preservative that was used very commonly in vaccines. The Food and Drug Administration in America was doing effectively what is, I'd like to call, housekeeping in the 90s. And it looked at everything and saying, well, we should be getting mercury out of food and drugs, and if we don't need it in there, we should take it out. And they reviewed the vaccine schedule, and they looked at it, and they basically saw that even with the vaccine schedule using thimersal-based vaccines, the amount of mercury didn't exceed their guidelines or the WHO guidelines. However, they kind of said, there's no evidence of harm, but we should probably take it out as a purely precautionary measure. Didn't quite go how they thought it was going to. The American public, being notoriously independently minded, thought that there was some kind of major cover-up. This was heightened by lots of adverse publicity around mercury in the 50s and 60s related to things like the Minamata Bay waste spillage. And so that, coupled with a rising number of vaccines in the vaccine schedule, and coupled with now a rising recognition and diagnosis of a condition known as autism, allowed people to start making links and said, actually, this thimersal-induced side effects in autism, this is the link. And that birthed in the late 90s the autism and thimersal, which basically is this group, this advocacy group called Safe Minds. And the problem is that Safe Minds had got some very serious and influential buy-in. You had congressional representatives, Robert Kennedy Jr., lawyers taking out adverts, basically saying, we'll pay you if you're an expert witness and you want to testify against medical establishment. And you had celebrities, Oprah Winfrey, etc., going on talking about the risk of thimersals. Don't forget there was no obvious risk found and hadn't been found to date. And then that gets us to Wakefield and the Lancet in the late 90s. And this was, as we're probably all very much aware, a consultant gastroenterologist at the Royal Free who had published multiple papers over the past kind of decade, which kind of started to look at measles and Crohn's, and then started to say, well, actually, no, it's the live attenuated vaccine that causes Crohn's, and culminated in a paper published in '98 about ileal-lymphoid nodular hyperplasia. I won't go into specifics, it's very well known. But essentially, it was claiming a link between MMR and autism through this enteropalytic new diagnosis. And the thing was that really set the world afire was he went off script. So there was a press conference at the Royal Free. The Royal Free were really keen to push this, so they were getting paid for it. And he sort of said, look, I can't continue to advocate the use of multiple combination vaccines until this issue has been resolved. And the paper really was looking at this very small case series of 12 children. And it's this. If you look at it now, you could argue that it's very childishly written and wouldn't stand up to scrutiny. But actually, the paper itself said there's no causal connection being proven. It was just interesting and further research was needed. But Wakefield, in his press conference to the national kind of press, said, look, the MMR should be suspended until we can show it's safe. He then left the Royal Free. A couple of years later, he's gone off to work in America, initially in the International Child Development Resource, and then he's gone into various advocacy groups. The effect on MMR in the UK was devastating. If you remember back very earlier in this talk, the reproduction number for measles was about 18. You need at least 90%, probably 95% coverage to give herd immunity to stop measles becoming endemic, and it plummeted. The uptake plummeted, and mothers lost all confidence, and that's still there today about vaccines. Is it safe? Can we give combination vaccines? Is it safe? And in the kind of great realms of Scooby-Doo, he would have got away with it too if it wasn't for those pesky kids. And the pesky kid we're talking about here is Brian Deere, who's the excellent Sunday Times reporter. And if you've never seen the episode he did in 2004, it's available on YouTube. Just watch it. It's fantastic. He did a lot of investigative detective work. He's written a couple of books on it. And he unpicked some really very interesting facts, that the 12 kids were not picked at random. They were pre-selected by a lawyer who was preparing a lawsuit against the MMR manufacturers. And these children were put forward by anti-MMR campaign groups. The Royal Free, who was hosting all this, was being paid by the Legal Aid Board to investigate whether there was a link, to basically allow Richard Barr, the lawyer, to do a class action lawsuit. Wakefield himself applied for a patent for a single jab measles vaccine that he didn't disclose at the press conference. He was also working on a kind of diagnostic kit for autistic enterocolitis. One of the people that worked with him in the lab couldn't find any evidence of measles virus in the children that were reported to have it. There was no ethical approval given for doing the colonoscopies. When they went back and reviewed the medical records, they were different from the paper. It is a tissue of lies. But what really is staggering is that the Legal Services Commission, who were looking into this, was paying Wakefield hundreds of thousands of pounds, and actually we don't actually know how much money he pocketed at the end of the day before he fled. But it's likely to be several £100,000 purely to do this work. As we know, there was a GMC tribunal. He denied all charges and still does, and thinks that he's done wrong. GMC was damning in its verdict. He was failed in his duties as a consultant. He was dishonest and irresponsible, and he was erased from the register. And it's one of the few times that the Lancet retracted, and actually the editor-in-chief really should have thought about their position at this point. But it took decades to repair the damage. There was no link between MMR and autism. There was no link between thiomersals. MMR don't contain thiomersals anyway, so that anti-vaxxer kind of claim was completely spurious. And this chart is slightly kind of complicated, but effectively the top line of the blue is the number of deaths of people who have not had measles vaccine versus the line at the bottom, the solid line, which is the number of deaths you get per million in children who have been vaccinated. And there is this clear gap that widened where people who were not having the vaccine were effectively allowing measles to start to run rampant. We would get outbreaks and epidemics. And we're still feeling the measles fallout to this day. Congo had a big outbreak a couple of years ago, which killed thousands, and you've got to remember that measles kills thousands. So obviously we do have the internet to consider here. Obviously that is allowing information to spread much more quickly than it was, say, 100 years ago, where it was pamphlets being handed out at rallies. Interesting facts that most of the vaccination websites in America are anti-vax, compared to, say, an equivalent country in North America, Canada. The majority of the American public still trust medical scientists on vaccines, but only just. So there's 45% of people don't trust medics on vaccines. And you get these public sentiments expressed by respectable and intelligent celebrities, such as our dear friend Donald Trump. And the problem is that even though we can look at that and go, it's complete nonsense, people believe this. People will believe what they think these celebrities are saying. So what can we do? Well, WHO has declared vaccine hesitancy and anti-vaxxer as a global threat, added to antimicrobial resistance and multi-resistant TB, etc. Governments are adopting legislation around the world to basically take fake news, false information and misinformation and disinformation down, although with variable success. Interestingly, if you look at the kind of legal routes that the anti-vaxxers have gone down, there's been many claims filed in the US court, the vaccine court, for autism related to vaccines. Only one had money paid out. 738 were dismissed. In February 2009, it ruled against 3 autism claims, proposing 3 different mechanisms. So effectively it's throwing out these mechanisms, and the 3 different judges found that there was no evidence linking those mechanisms for vaccines to cause autism. So in the kind of legal sense, we are winning the argument, but obviously the hearts and minds is still a long way to go. It's still going on and it's still affecting us to this day. We all remember the AstraZeneca issue in Europe. German newspaper published this article, hardly effective. It was rebuffed, but it came at the time when there was a lot of political flame going on about unfilled vaccine shipments. And you had, again, political leaders, influential public figures such as Macron saying, this wasn't effective for people over the age of 65, even though the European Medicines Agency didn't say that. It said the data was limited. And what it meant was that several countries didn't use it. And so they delayed their vaccine rollout. So how many people have died because they weren't vaccinated in a timely way? And again, a lot of this was politically driven. The subsequent issue around blood clots related to AstraZeneca led to several countries stopping completely, even though the actual rates of blood clots are infinitesimally small, about 15 per million after the first dose. And there's widespread skepticism about the AstraZeneca vaccine in Europe based on this publicity at the time. So anti-vaccination, hopefully you can start to appreciate now, it's a complex web of interconnected drivers. It's about personal liberty, the libertarian group. Anti-establishment, you have conspiracy theories coming in. I haven't really got into the Bill Gates microchip, but that's there. It's about people advocating for children's rights. There's a bit about religion now, but that's much less. And then there's this financial gain. People want to publicize something so that people nowadays, especially with kind of YouTube and some of these online entertainments where you have to click and you get ad revenue, there is lots of incentives that people need to understand. And vaccines do cause potentially serious complications. However, the problem we have compared to say that and other drugs is that vaccines are normally given to healthy people to keep them healthy, and they're often mandated. And the final thing to say is that vaccines have been so successful that the actual diseases that they protect against have kind of faded from public memory. And so that's allowing heuristic thinking to become faulty around the risks and benefits. If you look at the anti-vax, they're always kind of the adverse effects of an unknown origin. They're increasing. They're kind of biological plausible. They often occur close to the time of vaccination, and they have these kind of terrible outcomes. And those terrible outcomes vary around the world. So in the US and UK, it's autism. Go to France, it's MS. Go to Nigeria, it's infertility, particularly male sterility. So these are kind of things that you need to kind of think about. It's, one size doesn't fit all, it's nuanced. But actually, nothing's changed. In the last couple of hundred years in the anti-vaccination movement, they think the actual kind of fundamentals haven't changed. Those who think it's ineffective, they get relatives who've suffered illness or death. They've got infringement of human rights, and those who are advocating alternative practice. And to kind of end with kind of a slightly light note, the one I particularly kind of derive amusement from is the anti-vaccination group in America, which is the Clear Minds, who funded a study about the vaccine autism link, and the study actually showed there was no link, and they got very upset about it. And so with that, I will end my show.
Well, thank you very much indeed. That was really tremendous, incredibly informative review of a contemporary problem, which is not all that contemporary. It's similar effects have been going on for many, many decades. Could I ask anybody who has questions, please, to put it on the chat line? I'm sure there are a number of issues which people like to raise. I've certainly got one or two questions, if I may, while people are beginning to put their questions on the chat line. Can I just ask one thing which struck me? That you were talking about the smallpox outbreak in the US related to laboratory changes, or
yes, the Qatar incident.
Yeah. One's immediate thought went to Wuhan and the laboratory, the biological laboratory in Wuhan. And I just wondered if you have any particular views. I mean, could this particular virus have leaked similarly to the smallpox virus in the US? Is it possible, or is it? Unlikely?
So the Qatar incident was polio. The Birmingham University with smallpox. We've had multiple releases. I mean, you can remember a few years ago the hand, foot, and mouth that was released from a laboratory in southern England that then went on to cause an outbreak in cattle. We know, so if we look at the facts, so we know that there's a Wuhan Virus Institute, which is effectively a coronavirus dedicated institute. It does do some other viruses, but the main thrust of its research is on coronaviruses. It was created in response to the first SARS. And one of its main aims has been to collect all of the coronaviruses it can from animals from around the world and actually genetically manipulate them to identify any potential kind of crossover event that may occur, mutations. So this is where it jumps from one species to another. Whether or not the virus emerged from there, I don't think we'll ever know for sure. Interestingly, there's parts of the genetic code for the receptor. There's kind of a fusion resign binding protein that is part of the receptor, is not present in a lot of the other coronaviruses. And so was it what we call kind of chimeric virus? We don't know. The various intelligence that's come out is being very kind of patchy, and unless China ever released information, which they won't, we won't know for sure. But it is biologically plausible. You know, it has happened before. Their locality, I mean, so the one that was doing the rounds in kind of micro circles, kind of slightly tongue in cheek, was that, you know, they had all these dead bats that they'd harvested for coronavirus, and rather than incinerate them, the bloke took them down to the wet market to sell. But I don't think we'll ever know for sure. But again, you know, it is biologically plausible that it has escaped from the research laboratory. It is a stunning coincidence, shall we say.
Thank you. Again, could I remind people just to put their questions on the chat line? Can I just ask you one thing which always intrigued me, and that is the actual vectors that have been used. I mean, the AstraZeneca has it's an adenovirus vector.
Yeah.
And the Pfizer is a different virus, a different vector.
Yeah.
Actually, what is the other vector? What is the vector for the virus?
So the viral vector effectively uses a non-pathogenic virus as its means. So effectively, you strip out the genetic material from within it. So in the case of AstraZeneca, it's a chimpanzee's adenovirus that has the ability to attach and adhere to respiratory epithelium in humans, but not go on to cause infection. So it's effectively an abortive infection. And what they do is they take it out and they manipulate the genes inside it so it produces the relevant protein that we want. The Pfizer and the Moderna are a completely different thing, where they are the naked mRNA, and then they're surrounded by a stable lipid membrane. And effectively what happens is you inject it intramuscularly. The lipid cell, you know, these kind of spheres of lipids then bind to the lipid cell membrane of muscle cells and antigen presenting cells and whatever else they can come into contact with, fuse and inject the mRNA directly into your cell, which then it gets moved to the Golgi apparatus. So it's effectively utilizing our machinery. So they're very cunning ways of doing it. The viral vector basically uses a virus to inject it, whereas the mRNA uses just pure RNA. The problem with the RNA viruses is obviously their stability, because they break down very easily. So if you shake them very hard, you'll disrupt all those lipid spheres. So they're much more fragile and have much harder kind of transport requirements.
That's fascinating. So a piece of mRNA is stuck into a piece of fat and injected into the muscle. So actually, we have got a question here. This is from Selena Master. And she says, I have just seen the latest James Bond film, which is based around a laboratory developing smallpox vaccine. Fascinating. Oh, I'm sorry you've told us the plot already. I'm looking forward to it.
I haven't seen it.
No, I haven't. I'm looking forward to seeing it. And she also breaking news today with a historic go-ahead for malaria vaccine to protect African children. So your presentation has perfect timing today. I would certainly agree with that. And another question here, what do you think about people who seem to think the Pfizer vaccine to be somewhat superior to other vaccines, like the AstraZeneca one? And that's an anonymous question. Is there any evidence for that? I mean,
so what we're now getting is what's called real-world evidence, and so what that effectively is looking at is infection rates. And the problem is that if you look at some of these numbers, the easiest way of kind of looking at just kind of raw data, you will see that the Pfizer and Moderna, the mRNA-based vaccines, have better effects. So they are more protective So instead of it being kind of 95%, they're 97%. But actually, instead of looking to see, well, you know, do they prevent you from getting symptomatic COVID? What we're really interested in is hospitalization rates, and they're both above 95% after the 2 doses. So it is kind of 6 of one and half a dozen of the other. Yes, you know, there will be, it's the Pfizer and the Moderna, because of their mechanism of action, where they basically utilizing our own machinery to just churn out the spike protein, will generate a bigger immune response. That takes longer to wane, so you see a better immune response from them. But obviously the thing that we cannot measure is the cell-mediated immunity bit, the T cells, and that still kind of juries out as to whether these different vaccine types are equivalent. Ultimately, I think the honest answer is that it doesn't really matter. I think it's quibbling over a couple of percentage points. Both of them are equally good at keeping you out of ITU. The advantage of the AstraZeneca has been and is its transportability, and actually, you know, we're not out of this mess until the world is out of this mess. And so until we get people in Africa, Southeast Asia, India fully vaccinated, there is always the potential for more viral mutations to occur and an escape strain to come out. And in that situation, the AstraZeneca is head and shoulders better than the Pfizer one, simply because you don't need ridiculous storage requirements. So yes, you know, it is slightly better, but actually clinically, I don't think it matters.
Fine, good. There's a question here from Graham Leah. Will there ever be some form of anti-common cold vaccine, or is that impossible with the various mutations of the viruses that occur many times per year?
Yes, so it's been the holy grail. So Portland Down in Salisbury Plain, which is now effectively our kind of reference laboratory for tropical diseases, was originally set up as a chemical weapons and biological warfare facility, and then in kind of the 50s and 60s was the Common Cold Lab, where they were trying all sorts of vaccines. And the problem that they recognized quite early on was the common cold was made up of about 20 discrete viruses. 4 of them were seasonal coronaviruses. You had metapneumoviruses, RSV, paraphilic, they had a lot. And it was impossible to find a universal vaccine or even a combination vaccine that gave you long-term immunity. As we've seen with this coronavirus vaccine, you know, the immunogenicity may not last very long. So I don't think we're ever going to get a common cold vaccine until we go down a completely different track for a common viral protein. And so, you know, don't give up your shares in Beecham's just yet.
Okay, well, look, thank you very much indeed, John. That really has been tremendous, and we're really grateful that you've taken the time to speak to us and present such a masterly presentation. It's really good, and I'm most grateful to you, and thank you very much indeed. John, I could, yeah, well, I have a few sort of housekeeping announcements to make, John, so please feel free to go, and again, once more with our grateful thanks. Thanks very much indeed, John. Yeah, I have a few announcements to make. Firstly, can I remind everybody about the AGM meeting on the 17th of November, which will take place face-to-face in the college. The details have been circulated, and I'm absolutely delighted with the response so far. Registrations have been really good. So if you've not yet registered, please consider doing so. Ed, do you have the slide to just remind people of the program?
It's up there, Irving.
Well, unfortunately, I don't see it, but in any event, we have an excellent program. I'm not sure whether anybody else can see the program. I cannot see it on my screen. But we've got an excellent program of speakers, and it'll be an opportunity to have a guided tour of the newly transformed college. Remember, it is a hybrid meeting. and will be screened online for anybody who prefer not to make the journey in person. Oh, there we are. So just to remind you, the excellent speakers, the picture there of the dining hall, which is in the Lumley Library. Really, I've been round the college already, and it's really very impressive. And the dining hall is extremely good, and I'm sure we'll have an excellent meal there. So please do register. If you can't, or you're worried about attending in person, please join us online. Just the next thing, this is of course the final one of our monthly Senior Fellow Society webinars, because we're now going to revert to our normal format of 2 meetings per year. The November meeting, which I've mentioned, and our summer meeting next year will be held in Bristol, and it's hosted by John Newman. I hope you've enjoyed the really wide variety of talks which we have had presented by superb lecturers, and I'm absolutely delighted that they've been so well supported. I'm most grateful to you all for your involvement. The live attendance for each talk has varied between 50 and 200 very often, 200 registrations, and in many cases several people have joined in with 1 registration, and also the recordings have been watched up to between 100 and 300 times. So it has been very popular. Just to remind you of the variety of topics that we've had, some medical, some historical, some COVID related, to some humanitarian issues. And as I say, all presented really by outstanding lecturers. Now, it has actually been suggested that we should perhaps continue with occasional webinars, and I very much appreciate your views on whether, in addition to our face-to-face meetings in November and June, there is a place for an additional 2 online webinars per year, probably in March and September. And so perhaps fellows could think about this, and I'll raise the topic again at the November meeting to obtain the views of delegates. But anybody who's got any comments before then, please feel free to contact me at any time to discuss them. Could I also remind fellows that we have vacancies on the Senior Fellow Society committee, and I do hope people will consider applying, putting names forward for these particular posts. I have to say that we're obviously anxious to attract as wide a diversity of individuals as possible, particularly in light of the recent report, the recent college's report on equality and diversity. So please give consideration to having your name put forward for inclusion within our Senior Fellow Society committee. Finally, obviously, I'm looking forward to seeing as many fellows and their partners as possible at the college in November. I have no doubt we will have an extremely entertaining day with a tour of the college, excellent talks, and a superb meal in very comfortable surroundings. So I do hope as many people as possible will consider joining us for our AGM. So until then, keep well, keep safe, and have a good evening. And thanks for joining the Senior Fellow Society webinar. Good evening.